The basic rhythm of breathing is controlled by respiratory centers located in the brainstem. This rhythm is modified in response to input from sensory receptors and from other regions of the brain. Your goals for learning are to understand how the respiratory centers control breathing to maintain homeostasis, to examine how PCO2, pH, PO2, and other factors affect ventilation, to understand the relationship between breathing and blood pH, to explore the factors which stimulate increased ventilation during exercise. Here's what you need to know: the basic principles of homeostasis, the general anatomy of the brain, the role of the inspiratory and expiratory muscles, the definition of pH, how to interpret the oxygen-hemoglobin dissociation curve. To review the breathing muscles or the oxygen-hemoglobin dissociation curve, click the appropriate link button. To see definitions of terms, click the bold red words. The control of respiration is tied to the principle of homeostasis. Recall that the body maintains homeostasis through homeostatic control mechanisms, which have three basic components: receptors, control centers, and effectors. Let's see how this applies to the respiratory system. The principal factors which control respiration are chemical factors in the blood. Changes in arterial PCO2, PO2, and pH are monitored by sensory receptors called chemoreceptors. The chemoreceptors send sensory input to respiratory centers in the brainstem, which determine the appropriate response to the changing variables. These centers then send nerve impulses to the effectors, the respiratory muscles, to control the force and frequency of contraction. This changes the ventilation, the rate and depth of breathing. Ventilation changes restore the arterial blood gases and pH to their normal range. The basic rhythm of breathing is controlled by respiratory centers located in the medulla and pons of the brainstem. Within the medulla, a paired group of neurons known as the inspiratory center or the dorsal respiratory group sets the basic rhythm by automatically initiating inspiration. Click the inspiratory center to see how it functions. The inspiratory center sends nerve impulses along the phrenic nerve to the diaphragm and along the intercostal nerves to the external intercostal muscles. Click the inspiratory center to see how it initiates inspiration. The nerve impulses to the diaphragm and the external intercostal muscles continue for a period of about 2 seconds. This stimulates the inspiratory muscles to contract, initiating inspiration. The neurons stop firing for about 3 seconds, which allows the muscles to relax. The elastic recoil of the lungs and chest wall leads to expiration. This automatic rhythmic firing produces the normal resting breathing rate, ranging between 12 and 15 breaths per minute. Click the inspiratory center to observe normal breathing. A second group of neurons in the medulla, the expiratory center, or ventral respiratory group, appears to function mainly during forced expiration, stimulating the internal intercostal and abdominal muscles to contract. In addition, other respiratory centers within the pons modify inspiration and allow for smooth transitions between inspiration and expiration. Their precise roles, however, are not fully understood. Although the basic rhythm of breathing is established by the respiratory centers, it is modified by input from the central and peripheral chemoreceptors. They respond to changes in the PCO2, pH, and PO2 of arterial blood, which are the most important factors that alter ventilation. Click the central chemoreceptors button to see their location. The central chemoreceptors in the medulla monitor the pH associated with carbon dioxide levels in the cerebrospinal fluid in the fourth ventricle. The chemoreceptors synapse directly with the respiratory centers. Now click the peripheral chemoreceptors button to see their location. The peripheral chemoreceptors are found in two locations: the aortic bodies within the aortic arch, and the carotid bodies at the bifurcation of the common carotid arteries. The peripheral chemoreceptors monitor the PCO2, pH, and PO2 of arterial blood. This information travels to the respiratory centers via the vagus and glossopharyngeal nerves. Click either button to review the location of the chemoreceptors. Let's look more closely at how the central chemoreceptors respond to carbon dioxide, the most important factor controlling the rate and depth of breathing. Click the central chemoreceptors to see their response. Carbon dioxide readily diffuses from the blood into the cerebrospinal fluid in the fourth ventricle. Here, carbon dioxide combines with water to form carbonic acid, which dissociates into hydrogen ions and bicarbonate ions. Most of the hydrogen ions within the cerebrospinal fluid are derived from this chemical reaction. The hydrogen ions stimulate the central chemoreceptors, which send nerve impulses to the respiratory centers in the medulla. Did you notice that as carbon dioxide increases, so does the number of hydrogen ions, which in turn lowers the pH? The central chemoreceptors actually respond to this pH change caused by the blood PCO2. Based on what you have just learned, see if you can predict what will happen to the breathing rate and depth if the arterial PCO2 increases. Click the correct answer. Correct! An increase in the PCO2 in the blood leads to an increase in hydrogen ions in the cerebrospinal fluid, decreasing the pH. The central chemoreceptors fire more frequently, sending more nerve impulses to the respiratory centers, which in turn send more nerve impulses to the respiratory muscles. This results in an increased breathing rate and depth, allowing more carbon dioxide to be exhaled, returning the blood PCO2 to normal levels. Click the blood vessel to see the flowchart again. Now, let's look at the role of the peripheral chemoreceptors. Increased carbon dioxide levels in the arterial blood result in decreased blood pH, which stimulates the peripheral chemoreceptors. They respond by sending more nerve impulses to the respiratory centers, which stimulate the respiratory muscles, causing faster and deeper breathing. More carbon dioxide is exhaled, which drives the chemical reaction to the left and returns the PCO2 and pH to normal levels. Click the blood vessel to learn how the peripheral chemoreceptors respond to other arterial pH changes not caused by carbon dioxide. The peripheral chemoreceptors also respond to acids such as lactic acid, which is produced during strenuous exercise. Active muscles produce lactic acid, which enters the blood, releases hydrogen ions, and lowers the pH. As we have just seen, the decreased pH stimulates the peripheral chemoreceptors to send more nerve impulses to the respiratory centers, which stimulate the respiratory muscles to increase the breathing rate and depth. More carbon dioxide is exhaled, lowering the PCO2 in blood, driving the chemical reaction to the left, and lowering hydrogen ion levels. Click the blood vessel to see the flowchart again. The peripheral chemoreceptors also monitor arterial PO2. However, the arterial PO2 must drop below 60 millimeters of mercury before the chemoreceptors respond. Click the graph to learn how the oxygen-hemoglobin dissociation curve explains this phenomenon. The normal alveolar PO2 of about 100 millimeters of mercury results in 98% hemoglobin saturation in the blood. If the PO2 drops to 60 millimeters of mercury, hemoglobin is still 90% saturated. As you can see from the graph, any increased ventilation in this range of PO2s results in only a small increase in the amount of oxygen loaded into the blood. Click the graph to continue. However, at very high altitudes, the alveolar PO2 may fall to 40 millimeters of mercury, and hemoglobin will be only 75% saturated. At this point, increased ventilation will make a dramatic difference in the amount of oxygen loaded into the blood. The low PO2 in the blood stimulates the peripheral chemoreceptors to send nerve impulses to the respiratory centers, which stimulate the respiratory muscles, increasing ventilation. More oxygen is inhaled, and the arterial PO2 returns to normal levels. Click the blood vessel to see the flowchart again. What changes will occur if a person hyperventilates, that is, breathes deeper and faster than necessary for normal gas exchange? Click the correct combination of buttons to indicate the changes in the blood that will result from hyperventilation. Refer to the chemical equation for help. Correct! During hyperventilation, carbon dioxide is exhaled, lowering the PCO2. This drives the chemical reaction to the left, decreasing the hydrogen ion concentration and increasing pH. Since the PCO2 is low, the central chemoreceptors send fewer impulses to the respiratory centers. Since the pH is high, the peripheral chemoreceptors also send fewer impulses to the respiratory centers, which send fewer nerve impulses to the respiratory muscles, thereby further decreasing breathing rate and depth and returning the arterial gases and pH to normal levels. Click the blood vessel to see the flowchart again. Now predict what changes will occur if a person hypoventilates. Hypoventilation occurs when the breathing rate and depth is too low to maintain normal blood gas levels. Click the correct combination of buttons to indicate the changes in the blood that will result from hypoventilation. Refer to the chemical equation for help. Correct! During hypoventilation, not enough oxygen is inhaled, so the PO2 decreases. In addition, carbon dioxide builds up in the blood, increasing the PCO2. This drives the chemical reaction to the right, increasing the hydrogen ion concentration and decreasing pH. The PO2 drops, but not enough to stimulate the peripheral chemoreceptors. The high PCO2 stimulates the central chemoreceptors to send more impulses to the respiratory centers. A decrease in pH stimulates the peripheral chemoreceptors, which also send more nerve impulses to the respiratory centers, which stimulate the respiratory muscles, increasing the breathing rate and depth. This allows oxygen to be inhaled, carbon dioxide to be exhaled, and drives the chemical reaction to the left, returning the arterial gases and pH to normal levels. Click the blood vessel to see the flowchart again. This chart summarizes how the three major chemical factors, PO2, pH, and PCO2, modify breathing rate and depth. Click a button to review each pathway. When the PO2 drops below 60 millimeters of mercury, the peripheral chemoreceptors send nerve impulses to the respiratory centers. The respiratory centers send nerve impulses to the respiratory muscles, increasing ventilation. More oxygen is inhaled, returning the PO2 to normal levels. When cells release acids into the blood, the acids release hydrogen ions, which lower the pH. This stimulates the peripheral chemoreceptors to send more nerve impulses to the respiratory centers. They, in turn, send more nerve impulses to the respiratory muscles, increasing ventilation. More carbon dioxide is exhaled, which returns the pH to normal levels. An increase in PCO2 leads to a decreased pH in the blood, which stimulates the peripheral chemoreceptors to send more nerve impulses to the respiratory centers. In addition, the increased PCO2 leads to a decreased pH within the cerebrospinal fluid of the fourth ventricle. This stimulates the central chemoreceptors to send more nerve impulses to the respiratory centers. The respiratory centers send more nerve impulses to the respiratory muscles, which increase breathing rate and depth. More carbon dioxide is exhaled, returning the PCO2 and pH to normal levels. In addition, several other factors influence ventilation. These factors include voluntary control, pain and emotions, pulmonary irritants, and lung hyperinflation. Click the buttons to learn more about each factor. Pain and strong emotions, such as fear and anxiety, act by way of the hypothalamus to stimulate or inhibit the respiratory centers. Laughing and crying also significantly alter ventilation. Dust, smoke, noxious fumes, excess mucus, and other irritants stimulate receptors in the airways. This initiates protective reflexes such as coughing and sneezing, which forcibly remove the irritants from the airway. By sending signals from the cerebral cortex to the respiratory muscles, we can voluntarily change our breathing rate and depth when holding our breath, speaking, or singing. However, chemoreceptor input into the respiratory centers will eventually override conscious control and force you to breathe. Stretch receptors in the visceral pleura and large airways send inhibitory signals to the inspiratory center during very deep inspirations, protecting against excessive stretching of the lungs. This is known as the inflation or Herring-Breuer reflex. Now let's look at the changes in ventilation during exercise. Ventilation increases during strenuous exercise, with the depth increasing more than the rate. Remarkably, it appears that changes in PCO2 and PO2 do not play a significant role in stimulating this increased ventilation. Although the precise factors which stimulate increased ventilation during exercise are not fully understood, they probably include learned responses, neural input from the motor cortex, receptors in muscles and joints, increased body temperature, circulating epinephrine and norepinephrine, and pH changes due to lactic acid. Click a button to explore these factors. Ventilation increases within seconds of the beginning of exercise, probably in anticipation of exercise, a learned response. It also increases because the motor areas of the cerebral cortex, which stimulate the muscles, also stimulate the respiratory centers. Circulating epinephrine and norepinephrine secreted by the adrenal medulla stimulates the respiratory centers. Lactic acid, produced by exercising muscles, is another stimulus. Proprioceptors in moving muscles and joints stimulate the respiratory centers, as does an increase in body temperature. Here's a summary of what we've covered. The basic rhythm of breathing is set by the inspiratory center, located in the medulla. Other respiratory centers located in the medulla and pons also control breathing. Chemoreceptors monitor the PCO2, pH, and PO2 of arterial blood and alter the basic rhythm of breathing. Carbon dioxide, reflected by changes in pH, is the most important stimulus controlling ventilation. pH changes due to metabolic acids also alter ventilation. Oxygen stimulates ventilation only when the blood PO2 is very low. Other factors, such as voluntary control, pain and emotions, pulmonary irritants, and lung hyperinflation, also play roles in controlling ventilation. The control of ventilation during exercise, while complex and not fully understood, involves multiple inputs including chemical and neural factors.